Friends, do subscribe to the channel, hit the bell icon for more such video updates. Today we are going to talk about biomolecules and carbohydrates in each exam. Biomolecules, when we talk about, we need to understand one word, that is cellular pool. All the components of cell, whether it is organic or inorganic, together forms the cellular pool. There are two types of biomolecules, micromolecules and macromolecules. Micromolecules include simplest units like glucose, amino acids, fatty acids, etc. Whereas the macromolecules includes carbohydrates, proteins, lipids, etc. These micromolecules, they are used for metabolic process, whereas the macromolecules, they are complex, stored in bodies. We need to understand one word called as metabolic reaction. It is anabolic reaction plus the catabolic reaction. Now, whenever we talk about anabolic reaction, it simply means going from simple to complex. That is, it's a make process. Example, photosynthesis, where glucose is prepared. Catabolic reaction is complex to simple. So it simply means it's a break process. Where example is respiration, where glucose is broken down into ATP. In this we are going to study carbohydrates, proteins, lipids, nucleic acid, enzymes. These are all biomolecules found in cell. Let's talk about carbohydrates. As the name suggests, it is carbohydrates. Carbo is for carbon and hydrate is for water. So hydrated form of carbon is carbohydrate. For example, C6H12O6 is glucose. When we look at it, it is 6 water molecule and 6 carbon together. Carbohydrates, these are synthesized by plants in the simplest unit, that is glucose. So we need to understand unit of carbohydrate is glucose. All the carbohydrates, they have carbon, hydrogen and oxygen, where hydrogen and oxygen ratio is 2:1. For example, C6H12O6, hydrogen and oxygen is 12:6, that is 2:1. But we need to understand one thing. Some compounds do not have hydrogen and oxygen ratio as 2:1, but still they are carbohydrates. It simply means that they do not follow the rule of being 2:1, but still they are considered as carbohydrates. For example, it is rhamnose. Rhamnose, the formula is C6H12O5 and digitoxose C6H12O4, where hydrogen and oxygen ratio is not 2:1, but still they are considered as carbohydrates. Some compounds, they have hydrogen and oxygen ratio as 2:1, but they are not carbohydrates. For example, formaldehyde HCHO and lactic acid CH3CHOH. When we look at these formulas of formaldehyde and lactic acid, hydrogen and oxygen is 2:1, but they are not included in carbohydrates. So we can say these are not carbohydrates, and the one which not follows rule are carbohydrates. There are three functional groups found in carbohydrates. CHO, it's aldehyde group. C double bond O is ketone group. If aldehyde group is present, sugars are aldoses. Ketone group, it is ketoses. They have two or more OH group. There are two types of sugar, basically simple sugar and complex sugar. When we talk about simple sugar, they are used for reactions. They are complex sugars, they are always stored in body. Used for reactions, we have example like glucose and fructose, stored in body, starch, cellulose and glycogen. All the sugars, they are joined by a bond. So let's take one example. There are two glucose molecules. These glucose molecules, they are joined to each other by a special bond, and the bond is called as Glyco-Siddik bond. This bond is formed by loss of water molecules, so it's a condensation reaction. When I draw this structure of glucose, here you will understand how the glycosidic bond formation takes place. Friends, we need to understand that simple sugar has a formula of CnH2nOn. So this is one glucose molecule having oxygen in the ring. Both water molecule is lost. One sugar will contribute OH group and other sugar is going to contribute the hydrogen. This is alpha ionomer and beta ionomer of glucose. So beta ionomer contributes OH and alpha ionomer contributes H, so there is a loss of water molecule. And finally what we get is a disaccharide, and they're joined by a bond. This bond, what we see, is glycosidic bond formed by loss of water molecule. Let's see classification of carbohydrates. There are three types of carbohydrates: monosaccharides, disaccharides, and polysaccharides. When we say monosaccharide, the word mono stands for one and saccharide is for sugar. Disaccharide, 2 sugar. Polysaccharide, many sugar. When we see monosaccharide, it is the unit, so it cannot be further hydrolyzed, but disaccharide can be further hydrolyzed to give only 2 units. Polysaccharides can be further hydrolyzed to give many units. The basic unit of sugar is monosaccharide, so it's a basic unit of complex sugar. Disaccharide, these are simple carbohydrates, and polysaccharides, they are complex carbohydrates. Monosaccharides, they are soluble in water, sweet to taste, crystalline in nature. Their crystals are seen. Disaccharides are also soluble in water, sweet to taste and crystalline in nature. But polysaccharides, they are insoluble in water, tasteless, and they are amorphous in nature, powdered form. There are two groups of monosaccharides, aldoses and ketoses. Aldoses have CHO and ketoses have C double bond O group. Example of aldose sugar, glucose, xylose. Example of ketose sugar, fructose, ribulose. In disaccharide, the two monosaccharides are joined to each other by a bond, glycosidic bond. We saw bond is formed by losing water molecule. Example, sucrose. Glucose plus fructose forms sucrose. Lactose, glucose plus galactose makes lactose. And we have maltose, which is formed by union of two molecules of glucose. So we say maltose is glucose plus glucose. So these are the three examples of disaccharides, 1, 2 and 3. When we talk about polysaccharides, there are two types of polysaccharides: homopolysaccharides and heteropolysaccharides. Homopolysaccharides, they have same units, whereas heteropolysaccharide, they have different unit. Example for homopolysaccharide is cellulose, starch and glycogen. They are made up of one type of sugar. Heteropolysaccharide example is hyaluronic acid, which is found in the ovum surrounding the corona radiata cells. Now based on number of carbon, we can say 3 carbon sugar, triose, 4 carbon sugar, tetrose, 5 carbon sugar, pentose, 6 carbon sugar, hexose and 7 carbon sugar, heptose. Example of 3 carbon is glyceraldehyde. Example of 4 carbon sugar is erythrose, 5 carbon ribulose, 6 carbon glucose, 7 carbon heptulose. How to write the formula? Just remember the carbon, hydrogen and oxygen. Carbon and oxygen is same, but hydrogen is double. So it is C7H14O7. What are the functions of carbohydrate? First, it provides energy in the form of ATP by breakdown of glucose. Lactose is found in animals' milk. Carbohydrates, they form structural components, like they form cell wall. Example, cellulose. It's a reserve food material. For animals, it is glycogen, and for plant, it is starch. Whenever we talk about the structures, we need to understand glucose, the formula is C6H12O6. So this way you can draw the structure of glucose, and it is very easy to remember, as the upper one is having CH2OH group. When we talk about fructose, it is again C6H12O6. So we need to understand that glucose and fructose, they are isomers. They have same formula but different structure. One having aldehyde group and another having ketone group. When we talk about disaccharides, for example maltose, it is having glucose plus glucose. So what we need to do, we need to draw two structures of glucose sugar and they are linked by a bond. So we show that bond as glycosidic bond and we need to balance the entire sugar. Now disaccharide, one more example is sucrose. For sucrose, it is formed by the union of glucose and fructose. So we need to add two sugars, one of glucose sugar and other one of fructose sugar. The simple general formula for simple sugar is CnH2nOn, and for complex sugar it is C6H10O5 the whole bracket N. Now friends, let's talk about disaccharides. There are two types of disaccharides, reducing and the non-reducing. The reducing sugars, they have free aldoses and ketoses, but non-reducing sugar, there is no aldoses or ketoses free. Example of reducing sugar is maltose and lactose, whereas for non-reducing sugar it is sucrose. Let's understand what carbohydrate speaks. We are polyhydroxyaldehyde or ketones classified into mono, oligo and polysaccharides held together by glycosidic bond, supplying energy and serves as structural constituent of cells. Friends, when we talk about polysaccharides, we need to understand that there are two types of polysaccharide, starch and cellulose. When both of them they react with iodine, in starch we see blue black color, but in cellulose we see no color change. Why is it so that there is a color change in starch and no color change in cellulose? Both are made up of glucose. So when we talk about starch, we need to understand that the starch, they are helical secondary structures, whereas when we talk about cellulose, their secondary structure is not helical. Therefore when we add iodine into starch, it binds to the branches of the helical structure, and in case of cellulose there is no branches. We need to understand one important word for Neet, that is inulin. So what is inulin? It's a polymer of fructose. Friends, let's try and understand what are proteins. So when we talk about proteins, these are long chain of amino acids. So what we need to understand, amino acids are the unit of proteins. The term protein was coined by scientist called Berzelius in 1830. Proteins are macromolecules with high molecular weight. For example, hemoglobin has the molecular weight of 68,000 Dalton. It is the most abundant component of cell. All the proteins, they are host specific. For example, in bacteria and in humans, bacteria they have more than 2000 proteins, and humans they have more than 1 lakh types of proteins. Proteins consist of carbon, hydrogen, oxygen and nitrogen. Sometimes the proteins, they also contain sulfur. When we talk about structure of proteins, they are made up of amino acids. So whenever we write one amino acid, we need to understand there are always two end free, COOH and NH2. COOH is carboxyl and also called as C terminal, and NH2 is amino and also called as N terminal. Both the amino acids, if you want to join them, you are going to join them by peptide bond. So how this peptide bond is formed? Again, it is formed by loss of water molecule, as we have seen in glycosidic bond. So two amino acids, they are written. So COOH is going to contribute OH and NH2 will contribute H. So there is loss of water molecule. So finally we get a bond, and this bond is called as peptide bond by the loss of water molecule. So whatever amino acids are left behind, After losing out water molecule, it is called as residue. So what is residue? Remainder of amino acid after removing water molecule. Depending on the number of peptide bond. One peptide bond, monopeptide, two peptide bond, dipeptide, more than two, we can say polypeptide. Let us have the amino acid chains, two different amino acid chain, and if you want to link both the amino acid chains, we need to link by disulfide bridge. Yes, this is where sulfur is used. So two or more polypeptide chains can be linked by disulfide bridge. When we try to classify the amino acids, we need to classify as two major type: essential amino acids and non-essential amino acid. Essential means not synthesized in a body and it is must required from outside. Non-essential, they are synthesized in a body. Example: arginine, histidine, isoleucine, leucine, methionine, phenylalanine, threonine, lysine, valine. The non-essential amino acids are glycine, alanine, serine, aspartic acid, aspargine, cysteine, glutamic acid, proline, and tyrosine. When we talk about amino acids, there are three types: acidic, basic, and neutral amino acids. I will be giving you a shortcut how to remember this acidic and basic amino acids name. Let's first see the neutral amino acids. They are glycine, alanine, valine, leucine, isoleucine, phenylalanine, tyrosine, tryptophan, serine, proline, glutamic acid, cysteine, methionine, threonine, aspargine. When we talk about acidic amino acid, the examples are glutamic acid and aspartic acid. When we talk about basic amino acids, they are arginine, lysine, and histidine. So if we remember acidic and basic, rest all is the neutral. Let's see some important points with respect to Neet exam. Glycine is the simplest amino acid with lowest molecular weight, and there is absence of asymmetric carbon. When we talk about tryptophan, most complex amino acid, contains indole ring. Next is methionine and cysteine. When we talk about methionine and cysteine, we need to understand they are sulfur containing amino acids. Only two amino acids having sulfur. Next we have proline and hydroxyproline. Instead of NH2 group, they have NH group. When we talk about NH, it is amino, and when we talk about NH2, it is amino. So we need to understand the difference between NH2 and NH. Next amino acid is isoleucine. Isoleucine has a two asymmetric carbon. We talk about tyrosine. Tyrosine gives rise to dopamine. It's a inhibitor. Melanin, pigment, thyroxine hormone, adrenaline or noradrenaline hormone. So these are produced by the tyrosine. Lysine and arginine are type of amino acids. They have more than one group of amino. They are basic in nature. So these are the important points to remember with respect to amino acids. When we talk about classification of proteins, we need to understand there are two major types of proteins: simple proteins and conjugate protein. Simple protein, only amino acids and their derivatives. Conjugated proteins, they are going to have simple protein plus non-protein part. So simple protein will be basically amino acid and non-protein part will be prosthetic group. Let's take the example of simple protein having only amino acid. It is histone that we see in coiling of DNA, zine or from maize. For conjugated protein, the examples are nucleoprotein, glycoprotein, chromoprotein, flavoprotein, phosphoprotein, and metal protein. Now we need to understand that nucleoprotein is nucleic acid and protein. Glycoprotein, they are found in carbohydrates. They have as prosthetic group. Chromoproteins are pigments, flavoproteins, FAD, Phosphoprotein is phosphate and metal protein is metal. Nucleic acid is found in viruses. Carbohydrates are found in membranes. Chromoprotein is hemoglobin. Flavoprotein, they are used in ETS. Phosphoprotein is found in casein of milk. Metal protein example is nitrate reductase. These are the examples we need to understand. The functions of protein. Number one, they are enzymes. All enzymes are protein, but all proteins are not enzymes. Some are hormones. Some are structural protein giving structure. Some are contractile proteins, transport proteins, and we have defensive protein. For enzymes, we can take example of amylase. Hormones, insulin, growth hormone, etc. Structural protein, we have keratin for hair and nails. We have elastin for muscles. Contractile protein is muscle. Myosin, transport. Hemoglobin supplies oxygen to cell, and myoglobin supplies oxygen to tissues. For defensive protein, we have antibodies, immunoglobulin. Now, we need to understand the proteins with respect to Neet exam. Watch till the end so that you can understand the video. When we talk about amino acids, how to remember them? Acidic amino acids. So, if you want to remember acidic amino acid, just remember gluacanti. When we talk about gluacanti, GLU stand for gluacanti. GLU is glutamic acid. Asa is for aspartic acid, and anti is acidic. So if you remember gluacanti, then it becomes very easy to remember acidic protein. Basic amino acids, it's aldehyde babe. It's aldehyde babe. It's ARLYHI babe. Aldehyde babe, it means R for arginine, Li for lysine, Hy for histidine, and babe is basic. So aldehyde babe. Rest all are neutral amino acids. When we talk about amino acids, they are basically zwitterions, and as you all know, zwitterions is the one which have positive as well as negative charge on the same side. So example for this is NH3CHRCOO minus, and this will be plus zwitterion. Let's try to understand amino acids. They have chiral carbon. What are chiral carbon? Single carbon having 4 different groups. Now when the R is replaced by H, it becomes glycine. R replaced by CH3, it becomes alanine. R replaced by CH2H, it becomes serine. This is how you can remember the amino acids. When we talk about proteins, they are primary, secondary, tertiary, and quaternary. When we talk about primary protein, it's one. Now we need to understand with respect to function. Primary will be having NH2, secondary NH, tertiary is N with triple bond, and we have globular protein. For primary protein, they have only long chain of amino acids, and for secondary protein, they have alpha or beta pleated sheets. Tertiary proteins, alpha and beta are held together, but for tertiary it is 3D. Quaternary, it is 2 alpha and 2 beta held together. When we talk about primary structure of protein, we need to understand they are sequence of chains of amino acids. Secondary protein structure, they have hydrogen bonds on the peptide backbone. And tertiary proteins, they are 3-dimensional folding pattern of a protein due to side chain interaction. Quaternary protein is hemoglobin, which is made up of more than one chain, so 2 alpha and 2 beta. Friends, for my next video, I am going to upload the concept of lipids and enzymes with respect to Neet exam. Do subscribe to the channel, give a like to the video if you have understood the concept. This is your Sunil sir. Thank you very much.